You know this moment. You have a cut on your finger. You reach for the brown bottle in the medicine cabinet. You pour. A white foam erupts from the wound. It stings. It fizzes. And somewhere in the back of your mind, a voice says, "That means it is working. The bubbles are the germs dying." That voice is wrong. The bubbles are not germs dying. They are telling you something else entirely, something that involves one of the fastest enzymes in your entire body. Here is what is actually happening. Hydrogen peroxide is a simple molecule. 2 hydrogen atoms, 2 oxygen atoms. H₂O₂. Think of it as water. H₂O, carrying 1 extra oxygen atom it doesn't know what to do with. That extra oxygen makes the whole molecule reactive and unstable, constantly looking for a way to get rid of its extra passenger and settle back into being plain water. At the concentration you buy at the drugstore, about 3%, it's a mild antiseptic. But at higher concentrations, hydrogen peroxide is not mild at all. It has been used as rocket propellant. Concentrated peroxide can ignite organic material on contact. The molecule is not picky about what it reacts with. When you pour it on a wound, the hydrogen peroxide immediately runs into an enzyme called catalase. Catalase is a protein whose 3-dimensional shape is precisely tuned to do one thing: grab a hydrogen peroxide molecule, break it apart, and release water and oxygen gas. The reaction looks simple on paper. 2 molecules of hydrogen peroxide become 2 molecules of water and 1 molecule of oxygen. But chemically, this is what's called a disproportionation reaction. 1 peroxide molecule is oxidized, losing electrons and becoming oxygen gas. The other is reduced, gaining electrons and becoming water. Catalase is the tool that makes both halves of that trade happen at once. Catalase does this using an iron-containing ring called heme at its active site, the same chemical group found in hemoglobin, the molecule that carries oxygen in your blood. But where hemoglobin gently binds and releases oxygen for transport, Catalase uses its heme iron to tear hydrogen peroxide apart at extraordinary speed. Biochemists studying Catalase's reaction kinetics have described it as operating close to the theoretical speed limit for an enzyme, a rate limited almost entirely by how fast the peroxide molecules can physically reach the active site, not by anything the enzyme itself is doing slowly. A single catalase molecule can process millions of hydrogen peroxide molecules every second. Picture a fire hose aimed at a single lit match. That's the kind of mismatch you're looking at. This is why even a tiny drop of blood, carrying only a small amount of catalase from your damaged red blood cells, produces a vigorous foam the instant peroxide touches it. You're watching one of the fastest chemical reactions in biology happen in real time, on your own skin. But this raises a deeper question. Why does your body have catalase in the first place? Hydrogen peroxide isn't just something you buy at a store. Your cells produce it constantly, as a normal byproduct of simply being alive. The main sources are your mitochondria, the power plants inside your cells, and your peroxisomes, small compartments that break down fatty acids. Both processes leak electrons that react with oxygen, and some of that reaction ends up as hydrogen peroxide. Peroxisomes actually get their name from this exact job. The word combines peroxide with the Greek word for body, soma, because these tiny structures were named specifically for handling peroxide inside the cell. Each peroxisome is packed with catalase, ready to neutralize hydrogen peroxide the moment it forms. It's a remarkably direct system. The very compartment that produces the hazard also contains the tool built to diffuse it. If this peroxide were allowed to accumulate, it would cause real damage. It would oxidize proteins. It would attack the fatty membranes that hold your cells together. Through a process called Fenton chemistry, it can generate hydroxyl radicals, some of the most destructive molecules in biology, capable of damaging your DNA directly. Left unchecked, your own metabolism would slowly poison you. Catalase is the defense. It converts hydrogen peroxide into harmless water and oxygen before any of that damage can happen. It isn't a side effect of your biology. It's a requirement. Nearly every oxygen-breathing organism on Earth, from most bacteria all the way up to blue whales, relies on catalase, or a very similar enzyme, to protect itself from the peroxide its own metabolism generates. A small number of bacteria get by without catalase specifically, using other peroxide-handling enzymes instead. But the underlying problem they're all solving is the same one your own cells face every second. The existence of this enzyme helps explain why the myth about bubbles took hold in the first place. When 19th-century doctors first started using hydrogen peroxide on wounds, they saw the foam and reasonably assumed it was evidence of some chemical battle against infection. The fizzing looked like cleaning. The sting felt like it must be working. The reasoning was intuitive. It was also wrong. So let's return to the cut on your finger. When you pour hydrogen peroxide onto that wound, you're flooding it with a reactive molecule your body already knows exactly how to handle. The catalase inside your damaged cells, in your red blood cells, your skin cells, your immune cells, goes to work immediately. The foam you see is oxygen gas, pure O₂, produced at a staggering rate by an enzyme that was already sitting there, waiting. The bubbles are catalase doing its job. They are not germs dying. Does hydrogen peroxide kill some bacteria? Yes. At sufficient concentration and contact time, peroxide is genuinely antimicrobial, and it's been used as an antiseptic for over a century for exactly that reason. But the catalase producing the foam you see comes primarily from your own cells, not from bacteria. The visual signal in front of you isn't telling you about germs dying. It's telling you about your own enzyme, doing what it evolved to do, at a speed most chemical reactions never get close to. And there's a cost to that. The same peroxide that reacts with bacterial catalase also reacts with yours, and it can damage your healthy cells in the process. Fibroblasts and keratinocytes, the specific cells responsible for rebuilding wounded tissue, are vulnerable to peroxide exposure. This is part of why most doctors today recommend cleaning wounds with soap and water rather than hydrogen peroxide. The bubbles are genuinely fascinating chemistry. They are not evidence that the wound is being cleaned. This video is about the chemistry behind that foam. For specific guidance on how to actually care for a wound, talk to your healthcare provider. But the next time you see those white bubbles form, you'll know exactly what's happening. You're watching an enzyme you carry in every cell of your body, one of the fastest biological machines ever discovered, converting a reactive, unstable molecule into harmless water and oxygen, on contact, in real time. The sting, the fizz, the foam. It was never germs dying. It's your own body protecting itself, and it's one of the most extraordinary chemical reactions you'll ever witness, happening right on the surface of your skin. This is reasonable wonder. Big questions, clear science.